Guys, Anna is here, and in this video I'm going to teach you everything you need to know about gene structure and transcription in a summarized way to get your top grades in A-Level Biology. And without further ado, grab a piece of paper so you can draw along with me, and let's get into it. So to understand transcription, it's always good to learn about the gene structure first. And to do that, you need to know the definition of a gene, which will be slightly different to the GCSE definition. So gene is a section of the DNA that codes for a protein or a functional RNA. And it's very important to include two of those parts, guys. So protein will be made from messenger RNA, which we discussed in the previous video. But we commonly forget that actually there are so many other genes that will be coded for functional RNA, including transfer RNA, tRNA, and ribosomal RNA, okay? Because there are also essential components to the cell. So as long as you learn this definition, guys, it will get you maybe 1 or 2 marks across your A-level exams. Okay, perfect. So let's continue. So to understand the transcription as a process, you need to understand the structure of the gene and learn of its component. So if you copy this DNA fragment, so I usually draw it as 3 rectangulars, and we're going to put gene right in the middle of it. And there are many genes in our genome. I think humans have about like 23,000 genes, and we haven't even discovered some of them. So it's important to kind of understand how does the cell know where the genes are, which need to be transcribed. So gene has a section at the beginning called a promoter, which will mark the beginning of the gene. And then there is a section called terminator that marks the end of the gene. So promoter basically is a sequence of DNA that marks the beginning of the gene, and it has many different components that are going to bind to it, primarily transcription factors. And transcription factors, they're proteins that will bind to the promoter. You don't need to know specific names for A-level, but if you are going to study biology at university, you will learn that there are different ones in prokaryotic and eukaryotic cells, and they'll bind in a different order. But for the sake of it, we know the transcription factors that will bind to the promoter, and what they do is that they will end up recruiting RNA polymerase, an enzyme that basically will synthesize messenger RNA. And it will travel along the gene, it will synthesize mRNA, and once it reaches terminator sequence, the RNA polymerase will dissociate, because then it knows, okay, well, it's the end of gene, there is no need to transcribe it any longer. And that's how the cell knows where the gene begins and where the gene ends. So using promoter and terminator sequence. So this brings us straight away to transcription. So we understand how we can find the specific gene. And then you guys need to remember that the transcription is the process of producing of messenger RNA from a gene. Okay, it could be different RNAs as well, but for A-level, you just need to know about the transcription of messenger RNA. And as in my DNA replication video, I want you guys to draw a DNA fragment. Remember, it's going to be very long. In cells, the DNA is about 2 meters each in the nucleus, but we can't draw that in our notes now. So I just want you to draw the DNA fragment, and I want you to draw the second structure. And notice that it's supposed to be the same fragment, guys, same length. I know I've done it a bit longer, but please don't worry about that. It's just kind of a quick sketch. But can you see there is this kind of structure that is opened up in the middle? And remember, same enzyme that occurs in DNA replication, there is a DNA helicase, and it's an enzyme that breaks hydrogen bonds between 2 complementary strands of the DNA. But because the cell has 2 meters of DNA normally, it's really inefficient to break the hydrogen bonds across entire 2 meters of DNA when the cell needs to only transcribe 1 gene, which might be 100 to 1,000 nucleotides. So actually, in the case of transcription, you might see these diagrams where it's only part of it, the DNA is going to be opened up, and we refer to it as DNA bubble. And once DNA bubble is open, so the hydrogen bonds broken down between the nucleotides, then the RNA polymerase enzyme can come and join in, and it will travel in one direction, and as it travels along the DNA, it will synthesize a complementary copy of messenger RNA, as we abbreviate mRNA. And remember, guys, it's RNA polymerase if we're producing RNA, so do not mix it up with DNA polymerase, which is part of DNA replication. And again, guys, for exams, you need to know the definition, which is usually going to be a 2-marker. So RNA polymerase joins 3 adjacent RNA nucleotides by formation of phosphodiester bonds by condensation reaction. So please make sure to learn this by heart. It comes up quite often in exams. Okay, and this is, guys, only 2 steps of transcription that you need to know. Obviously, it's a much more complicated process in reality, but this is everything you need to know to get your top grade in biology, which is not too bad. And the last thing you need to know is that this process happens in the nucleus, not in the cytoplasm, but in the nucleus. So add this to your notes. And in our next video, we're going to go through how the RNA undergoes splicing, and then we're going to move on to translation. And thank you so much for watching this video. I hope if you have any questions, then please do send me a message. Press the thumbs up, subscribe to my channel, and if there are any particular topics that you really want me to cover before your exams, then please let me know down in the comments below, and I'll see you in my next video. Thank you very much. Goodbye.